Hello everyone, my name is Mark Noonan, and today we are going to be looking at a few hormones that are produced within the cell, and we will also be looking at the mechanisms by which they are produced. I will show you via a flowchart, and we'll describe how they are produced. Alright, so the first molecule we're going to look at here is aldosterone, and that is a steroid hormone secreted by the adrenal cortex. It sits right on top of the kidneys, and its function is to reabsorb sodium and water in order to raise blood pressure. So given that it is a steroid hormone, we know that some of it is derived from cholesterol, which is a lipid. So here we have cholesterol being made in the smooth ER, so that's the structure right here. And then remember, the smooth ER lacks ribosomes. And from there, from the smooth ER, there's actually quite a few different transport processes that take place between the smooth ER and the mitochondria. So the first one is transported to the inner mitochondrial membrane, where it is transformed into pregnenolone. From there, back to the smooth ER, where it becomes a molecule known as 11-DCS. And lastly, back to the inner mitochondrial membrane, where it is transformed into our final product, which is aldosterone. And aldosterone, because of its polar nature, is able to smoothly transition and diffuse out of the plasma membrane. And from one of the articles that I read, it is thought that these actin filaments here are what is involved in the transport within the cell and help it get from the smooth ER to the mitochondria. And when it's in the bloodstream right here, in order to increase the half-life of the hormone, it is bound to a transport protein. And from there, it is the target cell is our cells in the distal convoluted tubule. And these cells are rich in mitochondria to ensure that ATP is present. And we need ATP to allow for the reabsorption of sodium. Alright, so the next hormone we're going to look at is follicle stimulating hormone, or FSH. And this is a protein hormone that is produced in the anterior pituitary, up here. So this is a glycoprotein, so that means we need two components to this. We need the glyco portion, or the sugar portion, and the protein as well. And as we learned, that proteins are assembled in the ribosomes, so I've drawn those over here. We have our free ribosomes, and remember the ribosomes that are on the rough ER. And then another component is the sugar group. So based on some of the articles that I read, there's a phosphate group that attaches right about here to the smooth ER. And with the binding of the phosphate group, sugar molecules are built and transferred to specific amino acid sequences, so they kind of work together in order to make that glycoprotein. And then through transport vesicles, they are then transported over to the Golgi apparatus right here, and they're modified by the Golgi apparatus. There are certain enzymes like glycosidases and glycosyltransferases that help transform this protein into a glycoprotein. And then preparing for export, secretory vesicles help FSH diffuse via exocytosis, where they then enter the bloodstream, which I have drawn right here. And then the primary target of FSH are Sertoli cells, which are found in the gonads. And then FSH enters these cells via G protein coupled receptors. And the FSH molecules act to stimulate ribosomal subunit S6 within these cells. Alright, and then lastly we have actin. Actin is one of our intracellular protein molecules. It is a microfilament that plays a major role in the cytoskeleton and is also involved in cellular movement. So as we talked about earlier, these molecules right here, that's our actin, and they are thought to be involved in the movement of aldosterone, which we had talked about earlier. So first, we will begin with the construction of our amino acids, which all happens in the nucleus. Our DNA is transcribed to form mRNA, and then mRNA is linked to a cytoplasmic ribosome. So remember, we have our two different ribosomes, our cytoplasmic or free ribosomes, and then we have the ones attached to the rough ER. But in this case, we're going to be attached to the free ribosomes, which help to encode a specific amino acid sequence. So next, in order to form our actin, there is a process called actin polymerization or nucleation, in which there are three specific actin monomers that make up these globular proteins, and then those globular proteins make up the actin molecules. And then when these three monomers make up the proteins, these proteins intertwine together they kind of twist together, and then they are located most of the time just inside the plasma membrane. So that's why I have them drawn right up here. And if you could see closely, I drew two different colors to show that they're intertwined. And then in the production of movement, which is one of the functions, actin interacts with myosin, and myosin is a motor protein, and that is what is responsible for the contraction of muscle cells.